Hi everyone, I'm going to try to give you a short lesson on DNA profiling. This is the assignment that I posted on Schoology. I'm going to be the new Bozeman for you guys. So I'm going to just run it straight through though, and if I mess up, whatever. I'm just going to improvise it right now. So for DNA profiling, let me pull the PowerPoint. The big idea with DNA profiling is this is how they would check, crime scene DNA, paternity, stuff like that. So big overview on the top, you see, you just collect the cells from the crime scene or whatever. You have to amplify the DNA. So this is through the process of PCR, the second row. The big idea of PCR is it just takes a section of DNA and it makes hundreds of copies of it. If you guys didn't do the Biozone worksheet, it goes into it in more detail. This is how they also check for, right now I mentioned like how they're testing for coronavirus. They'll take a little sample from the patient and then they put in the machine. They have a little section of the COVID-19 sequence, and then they try to see if that patient has some of that in them. After that, then they will do two things. They can either do gel electrophoresis, or they can do something called capillary electrophoresis. It's the same idea, but capillary electrophoresis, it runs the data through a computer, so it's like faster and more accurate. And then through that, then you compare with your samples and see if they match. So the worksheet that's online gives you an idea of how capillary electrophoresis works. So I'm just gonna try to highlight some of the key ideas. Okay, so the first thing is STR stands for short, tandem, repeat. Basically, in our DNA, we have these sections that they're non-coding. So most of our DNA is the same, but we have some sections that would vary. And so these are called the non-coding regions, and it doesn't really matter if it varies because this isn't parts that code for a protein. So this is an example. This guy on the top says the short tandem repeat is the sequence AGAT, and each of these little rectangles represents a repeat of that. The person below that, they have 4 repeats of it, and then the guy on the bottom has 12 repeats. So that, if you ran it through a electrophoresis, either DNA gel electrophoresis or the capillary one, that's what will give you your DNA fingerprint. So these STR sites, the bigger the sequence is, for example, this one right here, 19 repeats, that will be higher up versus smaller pieces would move quicker through the gel. Let me show you on the next one. So gel electrophoresis, basically it separates DNA by size. The way it works is you take the DNA fragments and you load them into these wells on the top. So it says DNA samples are loaded into the wells using a micropipette. You run the machine, the power turns on, and the DNA fragments migrate through the gel. So DNA is negatively charged because of the phosphate groups. So when you turn the electric current on, the negative charged DNA will try to go to the positive end, which is at the bottom of the gel. And then when you turn the machine off, then you'll get a pattern like this. The largest pieces are up top, they move slower. The shorter pieces move faster. Just think about, actually never mind, I'll explain that to you when we come back. But basically the larger pieces, it's just harder for it to move through the gel, so they don't move as fast. And the smaller pieces go down further. So when you do this, if you go back to that first diagram, this chunk right here, oh wait, this chunk, this 19 repeat chunk is bigger than this smaller chunk. So if you look at the gel, the larger pieces, this BP stands for base pairs. So the larger pieces are closer to the top. The smaller pieces, 100 base pairs on the bottom, they move down further. So if you were to run this sample DNA, this chunk with 19 repeats would be the biggest chunk. And so that would be closer to the top, maybe it's this one. And the smaller piece would be closer to the bottom. And that's how you get your DNA fingerprint. With the assignment though, they use something else. They use something called capillary electrophoresis. It's same idea. It also separates DNA fragments by size, but in this case, they use a machine and a laser to detect the fluorescence of the pieces. And then you get a printout like this. So the way you read this is you have the number of base pairs on the top. You have this, this pattern on the bottom, which they call them standard markers. It's letting you compare to something, so you know how to compare the lengths of the fragments. It's kind of like this marker. It's like a known ladder. We call it a known DNA ladder. With, we know this piece is going to be 1,200 base pairs. We know this one's going to be 1,000 base pairs, so that we can compare the other fragments. So like this one, sample B, this would be, I would predict this fragment is 800 base pairs because of this marker. So it's kind of like this on the bottom right here. In this diagram, the way you would read the data is if allele 1 has 5 repeats and allele 2 has 11 repeats, you would have a peak right here, and they actually like will mark it 5 repeats, and then a peak right here, and they'll mark it 11, meaning 11 repeats. And just a recap of before, alleles is just different variations. So this person got a chromosome containing 5 repeats from maybe one parent, and then the other one from the other parent, 11 repeats. But you could have 6 repeats, 7, 8, 9, 10, 11, it just depends. But in the end though, they're only gonna have two total alleles because they only have 2 chromosomes, or 1 pair. So the way you would look at this, it's similar to the gel where 100 is the biggest piece, highest number of base pairs. 10 right here would be the lowest. So it's equivalent to just taking this and like kind of shifting it vertically. So this 100 would be like the pieces closest to the well. And the ones at the bottom, 10 on this end, would be the pieces, the smallest pieces. So it's the same idea, it's just using a computer to kind of like print it out for you. So on the activity, they're gonna ask you to analyze a person's DNA profile, and then you figure out if it matches to the data. So one other thing is, because you only have, well, we have 2 chromosomes, one from one parent and one from the other, you have, you could be heterozygous or homozygous. So in this case, heterozygous would mean if these are your 2 chromosomes, the first one has 6 repeats, so TCAT, 6 times. That's this peak right here. The second one has, I guess they say 3.2 because it has 3 and then 2 bases here. So in this case, this person is heterozygous because the 2 chromosomes that they have different numbers of STRs, versus on the homozygous one, you would only have 1 peak because in this case, both of their chromosomes have 5 STRs. So the paternal allele, the maternal one, they both have 5. So if you were to look at it on a computer, it's just going to be 1 peak. And if you see it, it's higher. It's like double the size as the heterozygous ones because there's like more fluorescence that the machine catches up, or the machine will see. So if you look at this person's result, there's 2 peaks. And if you look up here, 5 repeats versus 11 repeats, this person would be heterozygous. But once again, this one, if they have 1 peak, it means that they had the same number of STRs from both their mom and their dad. Here's another example of what it could look like. So this is blurry, but whatever. The M and the P, maternal, paternal. So if you look at the maternal sequence, it's 8 repeating units of TATC. For the paternal, it's also 8. So if you look at what the printout would look like, it would have 1 peak at 8. So they are homozygous, versus on the side, maternal and paternal. The maternal has 10 repeating units versus the paternal has 9. So on the printout, you'll have 2 peaks, one at 9 and one at 10. So when you do your assignment, they're going to give you something like this, and they're going to ask for you to analyze that person's DNA to count up how many STRs they have, and then see if it will match the printout. Okay, the last part, the last page on the worksheet is an extension, which I still want you to do, but it's going to ask you to calculate using an equation. So if you get there and you have problems, just ask me. You can leave it on the discussion, or you could just message it to me. Okay. All right. And if you haven't already, watched the boson video first, or watch it again if you need to, because he kind of goes over it as well.